Okay, so for today's lecture, we are going to look at chapter 5, which is on cellular respiration. So basically, so this is the overview of chapter 5. Basically, it is the chapter that covers on cellular respiration. So as you know, the function of cellular respiration is to produce ATP, and the fuel for cellular respiration is mainly glucose. Okay, mainly glucose. So cellular respiration will break down glucose until it produces ATP molecules at the end of the process. Okay, so cellular respiration, it involves redox reaction, and then there are 4 stages of cellular respiration, which is the first one is glycolysis. um, okay, so this is the first stage. So glycolysis, for each of these 4 stages of cellular ten reaction.
Madam,
Okay, so kalau saya terputus, you all just type dekat chat box tu. Sebab saya tengah buka desktop juga. Okay, so then I know that saya terkeluar daripada meeting. Okay, so terus letak dekat chat box tu kalau saya terkeluar. Tak ada saya cakap sorang-sorang. Okay.
okay, Madam.
Okay. Okay, so basically, I'm going to re-explain on glycolysis. Okay, so for glycolysis, it has 2 phases, which is the first phase is the energy investment phase, and the second phase is the energy payoff phase. So in the why do we call it as the energy investment phase? Because during this phase, the stage or the phase will use ATP molecule. So how many ATP molecule is used during this phase? Is two. Okay, two ATP molecules is used during the first phase. And then for the second phase, why do we call it as the energy payoff phase? Because in the second phase, 4 ATP is produced. So for glycolysis, you have to count how many the net production of ATP molecule. So berapa jumlah ATP yang dihasilkan. So multiply so like so ATP and produced during glycolysis. Okay, so the next thing that you have to know the location of where glycolysis occurs. So glycolysis occur in the cytoplasm. Okay, in the cytoplasm. So the next stage is the formation of acetyl-CoA. So for this stage, it has several names. Okay, so based on what books you are referring to. So so another term for it is that you can also call it as pyruvate oxidation and also link reaction. So why do we call it as the link reaction? Because it links between the glycolysis stage and also the citric acid cycle stage. Okay, so glycolysis, it occurs in the cytoplasm. Citric acid cycle, it occurs in the matrix of mitochondria. So that's why it is called as link reaction. You can also call it as link reaction. So you can also call it as the pyruvate oxidation, because in this stage, the pyruvate, which is the product of glycolysis, will enter into this stage and will be oxidized. Okay, so once pyruvate is oxidized, it means that another molecule will be reduced. So what is that molecule? So the molecule reduced in this stage is NADH. Okay, NADH molecule. So NADH molecule, it is a very important molecule for cellular respiration, because the NADH molecule will function as the electron acceptor or electron carrier. So it carries electron to the last stage, which is the oxidative phosphorylation stage. Glycolysis also produces NADH. Okay, another product of glycolysis is that it also produces NADH molecule. Okay. And then the next stage is citric acid cycle. For the second stage, it has three reactions. Okay, three reactions to occur. For citric acid cycles, which occurs in the matrix of mitochondria, it uses the product of link reaction. So link reaction will convert pyruvate into another molecule. We call it as acetyl-CoA. Okay, acetyl-CoA. Acetyl-CoA will enter into the citric acid cycle, and then the citric acid cycle, as you know, it occurs in the matrix of mitochondria, and then the function of the citric acid cycle is to produce also NADH. Can citric acid cycle products are NADH. It also produces ATP molecule, and it also produces another molecule. We call it as NADH2. Okay, NADH2. So these are the important products of citric acid cycle. So these two molecules, NADH and also FADH2, will enter into the last stage of cellular respiration, which is oxidative phosphorylation. So oxidative phosphorylation, it has two components, which is the first one is the electron transport chain, and the second one is the osmosis. So electron transport chain, it consists of several electron carriers that are embedded in the inner membrane of mitochondria. Okay, so here you have the mitochondria. The mitochondria has two membranes. Okay, the outer membrane and the folded inner membrane. So the folded inner membrane, we call it also as the cristae. So all the electron transport chains are embedded in the cristae of mitochondria. Okay, so these electron carriers will carry electron from one electron carrier to another electron carrier. Okay, until at the end it is received by oxygen. So oxygen will function as the last electron, the last electron acceptor. Okay, oxygen will receive the electrons, and then oxygen also will combine with hydrogen ion. Okay, and then it forms water molecule. Okay, so as you know, the byproduct of cellular respiration is water. So water is produced during this stage. Okay, and then where is the source of these electrons? So the source of these electrons are from the NADH and also FADH molecules that are produced during glycolysis, produced during Link reaction, and also produced during citric acid cycle. Okay, so these are important electron carrier that will donate their electron to the electron transport chain along the cristae. Okay, so as the electron is transferred from one carrier to another carrier, the electron releases energy. Okay, it releases energy. So this energy will be used in the chemiosmosis reaction. Okay, so what is chemiosmosis? Chemiosmosis, it involves the energy which is released by the electron to pump hydrogen ion. Okay, to pump hydrogen ion from the matrix, okay, from matrix into the intermembrane space. Okay, into the intermembrane space, and you have to take note also the concentration of hydrogen ion in the matrix is low compared to the concentration of hydrogen ion in the intermembrane space, which is high. Okay, so that's why energy is used to pump hydrogen ion from low concentration to high concentration. So it is a form of active transport. Okay, so when you have a high concentration of hydrogen ion in the intermembrane space, what happens is that the high concentration of hydrogen ion will diffuse back into the matrix using protein channel. We call it as the ATP synthase molecule. Okay, so ATP synthase molecule, it has two roles, which is one as protein channel. Okay, channel. Why? Because it allows hydrogen ion to pass through it to diffuse to use the protein channel to diffuse into the matrix, and then at the same time the ATP synthase will synthesize ATP. Okay, so this is what happened during chemiosmosis. It uses energy from the electrons to allow hydrogen to move, okay, from to be pumped from the matrix into the intermembrane space and then back into the matrix. Macam tu. Okay, so these are basically the overview of what occurs, okay, during cellular respiration. So these are the topics of cellular respiration. So one, you have to know what is ATP, okay, and then you have to know the redox reaction that occurs. So what molecules is oxidized, what molecule is reduced, okay, and then you have to know that cellular respiration, it has 4 stages. Glycolysis is the first stage which occurs in the cytoplasm. And then you have the pyruvate oxidation, which is the second stage of cellular respiration, which occurs in the matrix of mitochondria. And then the third stage is citric acid cycle, which also occurs in the matrix of mitochondria. And then the last stage is oxidative phosphorylation, which involves the ETC, which is embedded in the cristae, and also chemiosmosis. Okay, so the only stage of cellular respiration that occurs outside of the mitochondria is the glycolysis, which occurs in the cytoplasm. Okay. So these are the learning objectives yang awak boleh baca sendiri. Okay, so ATP. So these are the basic. Okay, so this is the structure of an ATP molecule. So what does ATP stands for? Adenosine triphosphate. Okay, used as the main energy source for metabolic function. So these are the component of ATP that you have to know. Okay, so it consists of a 5-carbon sugar, which is ribose. Okay, which is attached to a base, which is adenine, and then which is ribose is also attached to a 3-carbon, a 3-phosphate groups. Okay, 3-phosphate groups. So if you look at the structure is similar to a nucleotide. Okay, so except for nucleotide, it only has one phosphate group attached to it. Okay, nucleotide has three, four more bases. You have the guanine, thymine, cytosine, and also uracil. For ATP, it only has adenine attached to it. Okay, so why is ATP capable of producing energy? So what is the use of that energy? So ATP, it is capable of um, releasing energy because when the third phosphate is released, the break this bond, okay, will release energy. Okay, it is an exergonic reaction. It releases energy. So this energy will be used for any cellular work. Okay, for any cellular work to be carried out. So what are the examples of this cellular work or activities? To catalyze chemical reactions, to move proteins from one region to cell, for active transport, can. So for endocytosis or exocytosis, all those activities within cells requires energy, and the energy is provided by the ATP molecule. So this is how the ATP molecule releases energy. So ATP molecule will undergo hydrolysis. It undergoes hydrolysis. So what does it involve? Hydro, it means breaking of bond using water molecule. Okay, so as you can see from the diagram, water is added into the reaction. Okay, so this is to break the bond between the second and the third phosphate. So basically the third phosphate is released. When the third phosphate is released, we call it as the inorganic phosphate. Okay, so when phosphate is released, if you see this symbol, I, the small I here, it stands for inorganic phosphate. So now, when the third phosphate is released, the ATP becomes ADP, adenosine diphosphate. It has 2 phosphate groups. Okay, so the reaction is exergonic. It means that it releases energy. Okay, so the amount of energy released is 7.2 kilocalorie of energy per molecule, per mole of ATP hydrolyzed. Okay. Okay, so the ATP cycle. So for ATP cycle, it involves both the catabolic uh, reaction and anabolic reaction. So what is catabolic reaction? Catabolism. It involves a metabolic pathway that breaks down a larger molecule into smaller molecule. And the process of breaking down larger molecule into smaller molecule, it releases energy. So in this case, energy from catabolism, okay, that releases energy, exergonic, involve what? Actually, the cellular respiration reaction that breaks down what? Glucose into smaller molecule, which is 2 molecule of pyruvate, and then into many molecules of ATP. So usually about 30 or 32 ATP molecule is produced. Okay, so the reaction releases the production of ATP. Okay, is from the energy released during cellular respiration. So glucose breaks down into pyruvate, and then pyruvate is used to produce energy. So the reaction is catabolic. Okay, from larger molecule into smaller molecule, releasing energy. Okay, so basically the energy released from the breaking down of glucose here is used, okay, to attach inorganic phosphate with ADP. okay, inorganic phosphate with ADP forming ATP. So this reaction releases energy, which is actually to form bonds, okay, to form bonds between phosphate, inorganic phosphate with ADP to form ATP. So this ATP molecule, in order for the ATP molecule to be used, okay, to be used to release energy, so it will undergo hydrolysis. Okay, here what happened? Hydrolysis occurs, okay, which is water is added, and then the breaking of ATP molecule, okay, to form back ADP releases energy. Okay, releases energy. So this energy is used for any activities within cells that requires energy. Okay, endergonic energy consuming processes. Any activities within cells that requires energy. Okay, activities within cells that needs or requires energy. So this energy comes from the hydrolysis of ATP molecule. Okay, for endergonic reaction within cell. Okay, energy requiring energy consuming processes. Okay, so catabolic pathway and production of ATP. So cellular respiration includes both anaerobic and aerobic respiration, but usually aerobic. Okay, so glucose is the main substrate for cellular respiration. So this is the normal reaction, as you know. Okay, for cellular respiration, it requires glucose. Okay, it requires oxygen. It produces carbon dioxide, water, and energy in the form of ATP, and also it produces heat. So this is uh, uh, the the need equation. The need, nampak cantik kan? The need equation of cellular respiration, but cellular respiration is not as simple as this equation. Okay, so it is not as simple as this equation. So cellular respiration actually, as we have looked before, okay, in previous slides, it requires 4 stages reaction. So the first stage, glycolysis, it involves 10 reactions. Okay, the second stage, which is the linked reaction, it involves 3 reactions. Okay, the third stage, which is citric acid cycles, it involves 8 reactions, and the last one, which is oxidative phosphorylation, it involves the movement of electrons and also hydrogen ions. So actually cellular respiration is not as simple as this equation as you see. Okay, so it involves many reactions to occur to produce ATP at the end. Okay. So basically you have to know, okay, bila masa glukos diperlukan, okay, bila masa oksigen diperlukan dalam reaction tu, bila carbon dioxide ni dihasilkan, bila air ni dihasilkan, dan bila ATP ni masa keempat-empat cellular respiration ni berlaku. Okay. Okay, so the next slide. So as you know, the main fuel for cellular respiration is monosaccharide, glucose. Kan? Glucose is the main substrate. But if your body is lacking glucose, such as when you are fasting, for example, your body can turn to other macromolecules to be used as fuel. Okay, so it can turn to fatty acid. Okay, the mitochondria can use fatty acid as fuel for cellular respiration, and but if there is no fatty acid, the body will find amino acid as the fuel. Okay, so by it is not as simple as that. So this molecule has to be converted into acetyl CoA through a series of reaction as well. Okay, so once this fuel, okay, is converted into acetyl CoA, then only the acetyl CoA can enter into the citric acid cycle, which is the third stage of cellular respiration. So the purpose of citric acid cycle is to produce NADH, and it also produce another molecule which is FADH2. Okay, kalau nak hasilkan NADH, dia daripada NAD plus, which is a cofactor or coenzyme. Tapi kalau FADH, dia tak ada plus sign, dia FAD sahaja. Okay, so in order to convert NAD plus to NADH, so this molecule has to be reduced. Okay, it has to be reduced. Same also for FAD. Okay, it also has to be reduced. So what does reduce mean? It will receive, these two molecules will receive hydrogen ions and also electrons. Okay, so then only it can be converted into NADH or FADH2. Okay, so ingat FAD tak ada plus sign. NAD saja, NAD plus ada subscript. Kalau exam jawab dia FAD ada plus sign, salah. Kalau tulis FADH saja tanpa the two subscript pun salah. Yeah, so remember that you have to know how to write this molecule properly. Okay, so NADH or FADH2, which is produced during citric acid cycle, will enter into the oxidative phosphorylation stage, which is to produce a large quantity of ATP. Citric acid cycle can also produce ATP, but the quantity is really small. For one cycle, it only produces one ATP. Okay, so really small ATP, really small amount of ATP is produced through a reaction, we call it as substrate level phosphorylation. Okay, kalau ini a large quantity is produced, okay, the process we call it as oxidative phosphorylation. Okay, kalau sikit it substrate level. So the same diagram as before. So amino acid can be fuel, sugar obviously, and also glycerol, fatty acid can be fuel as for cellular respiration, but this molecule has to be converted first, okay, to another molecule which is acetyl-CoA. Okay, so for sugar, when it enters in the first stage of cellular respiration, which is glycolysis, which occur in the cytoplasm, so glucose will be broken down into two molecules of glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is a 3-carbon sugar, okay, which is produced from the breakdown of glucose molecule. Glucose, as you know, is a 6-carbon molecule. So nampak glucose, one molecule, it has 6 carbon. But when it is broken down into 2 molecules, so the 2 molecule will only have 3 carbons. Okay, so this is glyceraldehyde 3-phosphate. And then the short name for it is G3P. So this G3P molecule will be into pyruvate. Okay. So pyruvate pyruvate produced at the end of glycolysis is 2 molecule. Okay, 2 molecule of pyruvate is produced at the end of glycolysis. So pyruvate is also a 3 carbon into the link reaction. Okay, link reaction. So the link reaction will convert pyruvate into acetyl-CoA. Acetyl-CoA is the 2-carbon sugar. So mana hilang? Asalnya dia masuk sebagai 3 kan? Tapi dia keluar sebagai 2-carbon compound. Mana lagi satu carbon hilang? Dia hilang sebagai carbon dioxide. So carbon dioxide is produced during link reaction. Carbon dioxide is also produced during citric acid cycle. So itu kena tahu, carbon dioxide produced during link reaction. Carbon dioxide is also produced during the citric acid cycle. So the of glycolysis, the product of link reaction and citric acid cycle, which are NADH and also FH2, will enter into the oxidative phosphorylation stage to produce ATP. Okay, so that is how cellular respiration occurs. So it depends on the cell. So some cell produces 30 ATP, for cellular respiration altogether, and some cell produces 32 ATP molecule. Okay, for the whole cellular respiration reaction. Okay. So next is substrate level phosphorylation. So a small amount of energy is generated by substrate level phosphorylation. So substrate level phosphorylation is it involves reaction that produces ATP. Okay, so how does ATP produce through this mechanism? So the ATP molecule is produced by transferring phosphate group from a substrate that has phosphate. So as you can see from this diagram, okay, so this is the initial substrate. As you can see, the green substrate here, it is attached to a phosphate. So when the substrate is attached to a phosphate, when a substrate is attached to a phosphate, it causes the phosphate to be highly energized. Okay. So, what happened in the reaction? So the substrate with phosphate attached to it will enter into a specific enzyme, and then the enzyme also have active site for ADP. Okay, so the phosphate from the substrate is then transferred to ADP. ADP will become ATP, and the product produced now has no phosphate. Okay, so that is substrate level phosphorylation. Substrate level phosphorylation occurs during glycolysis and also Krebs cycle. Okay, Krebs cycle is another name for citric acid cycle. Okay, Krebs cycle, citric acid cycle is another name for citric acid cycle. So oxidation and also reduction reaction. So some molecule will be oxidized and causes another molecule to be reduced. Okay. Sekejap ya. Sekejap. Okay, kita sambung. Dengar suara saya?
Dengar, Madam.
Madam.
Dengar, Madam.
Okay, sebab dia lagging tadi. Okay. Okay, so for redox reaction, so some molecule in the reaction will be oxidized and another molecule will be reduced. Okay, so for respiration, the oxidative phosphorylation stage will produce the most ATP. Okay, so for glycolysis, it only produces a net of 2 ATP. Okay, a net. So fahamkan kenapa saya cakap net tu, right? And then for link reaction, no ATP produced. Okay, for Krebs cycle or citric acid cycle, for one cycle, for one turn, it only produces one ATP. Okay, one ATP. But for oxidative phosphorylation, it produces the most amount of ATP molecule. So nanti kita tengok berapa banyak ATP yang terhasil semasa oxidative phosphorylation. So as you know, it involves the transfer of electron from one substrate to another. And then glucose, if you look at in this reaction, glucose is oxidized to carbon dioxide. Okay, so oxidized, it means that it loses electrons. Okay, glucose will loses electrons and also hydrogen ions. So nampak from glucose that has so many hydrogen atoms. So at the end, it only becomes carbon dioxide. So it is oxidized. It releases hydrogen ions and also electrons. As for oxygen, okay, so oxygen is reduced. Reduce maksudnya dia akan terima hydrogen ions and also electrons. Okay, hydrogen ion and also electrons to become water molecule. Okay, so glucose is oxidized, oxygen is reduced. Oxygen gains electrons and also hydrogen ions from glucose, and in the process, energy is also released during cellular respiration. But remember, cellular respiration is easy as this equation. Okay. So electrons or hydrogen atoms are not transferred directly to oxygen but usually pass to an electron carrier which is NAD+. Tadi saya dah cerita pasal NAD+ kan. So NAD+ function as electron carriers. Okay, so it is reduced because it receives hydrogen ions and also electrons to become NADH. Okay, which will feed the oxidative phosphorylation stage. So NAD+, so the name is nicotinamide adenine dinucleotide. So this is the structure of the NAD+ molecule. So if you look at the name of the molecule, dinucleotide. Okay, so if you look at the structure, it actually looks like two nucleotides attached together. A base, a 5-carbon sugar, and a phosphate. A 5-carbon sugar, a phosphate, and this one it replaces the base, which is the nicotinamide part. Okay, so the nicotinamide adenine dinucleotide. Okay, so that is NAD+. Functions as the coenzyme. Okay, coenzyme. Coenzyme, it means that if you remember in chapter 4, enzyme, okay, so requires a cofactor in order for the enzyme to be activated or to function. Okay, so NAD+ is a coenzyme. For enzyme, dehydrogenase. Okay, so dehydrogenase. So if you look at the diagram, the name, dehydrogenase, what is the function of this enzyme is to remove hydrogen atoms from a molecule. Okay, so this is hydrogen atom. Okay, so in hydrogen atom it has what? Hydrogen ion, so electrons. Okay, electron. So so NAD+ is a coenzyme for the enzyme dehydrogenase. The function of dehydrogenase is to remove hydrogen atom from a substrate. Okay, so nampak. So bila dia remove, so the hydrogen atom will the components are tribes. So nampak dia akan jadi NAD plus dan plus H plus electron. Macam ni lah NAD.
Madam, suara madam tak clear.
Putus ke?
Putus.
Saya nak tukar. Dah daripada slide mana?
Saya dari slide
Slide yang
yang
NAD+.
baru ni.
Slide yang pasal coenzyme tu. Okay, saya tukar mic desktop. Dengar?
Ya, madam.
Dengar, madam. Okay. Ini mic desktop, presentation saya dekat laptop. So laptop guna wifi. So laptop akan ada tendency untuk dia ter-remove. So kalau saya ter-remove, awak bagi tahulah. Ya? Suara saya akan dengar sebab dekat desktop disambung kabel dia more stable. Tapi kalau dekat laptop saya yang saya tengah present ni, dia guna wifi. Okay, so wifi akan disconnect lah. Dia akan ada tendency untuk ter-disconnect. So kita akan, so kita sambung balik semula pada slide yang ni. Okay, so the enzyme dehydrogenase. So as I've mentioned, so what is the function of enzyme dehydrogenase? Remove a pair of hydrogen atoms, okay. So the hydrogen atom, it removes 2 hydrogen atoms, okay. So the 2 hydrogen atoms will become 2 hydrogen ions and 2 electrons, okay. 2 hydrogen ions or 2 protons and also 2 electrons from the glucose. Glucose di sini dia menggambarkan daripada apple lah. The apple, the fruits or the food that you eat obviously contain glucose. That is the fuel for cellular respiration. So the glucose will be reacted, okay, by the enzyme dehydrogenase that removes hydrogen atoms. So it becomes hydrogen ions and also 2 electrons, and then delivers these 2 component to NADH. NAD plus. So NAD plus will be reduced to NADH. Okay. So tengok kat sini satu je kan? Satu je NADH. NAD plus. A plus 2 hydrogen ion plus 2 electrons. It becomes 1 NADH. Okay, and H plus. So this H plus ion is released to the surrounding solution. Okay, so this hydrogen ion is released to the surrounding solution. So nampak satu hydrogen ion akan NAD plus ambil, 2 elektron ni akan masuk lah kan. Satu dekat hydrogen ion yang dia terima, satu yang dekat plus sign here. Okay, so it becomes NADH. Okay. Okay, so next, the 4 stages of cellular respiration. So I've mentioned all this. Okay, so first stage is glycolysis. It occurs in the cytosol or cytoplasm. It breaks glucose, a 6-carbon sugar, into 2 molecules of 3-carbon sugar, which is pyruvate. Okay. The second stage is link reaction or formation of acetyl-CoA or pyruvate oxidation. So it occurs in the matrix of mitochondria. Boleh tambah kat sini. Matrix of mitochondria. And then, so for this stage, it converts pyruvate into acetyl-CoA. Pyruvate is a 3-carbon compound. Acetyl-CoA is a 2-carbon compound. So it means that during this stage, carbon dioxide is released. Okay. So the third stage is citric acid cycle. So the citric acid cycle also occurs in the matrix of mitochondria. So during this stage, it produces NADH and also FADH2 and also ATP. Okay, ATP. So the last stage is oxidative phosphorylation. Citric acid cycle also produces carbon dioxide as byproduct. Okay, so for the last one, oxidative phosphorylation, which involves the actual synthesis of ATP molecule. So this stage involves the electron transport chain and also chemiosmosis. So this one, it involves the ETC is embedded in the cristae. Okay. Cristae. So chemiosmosis, it involves the movement of hydrogen ions, okay, across the intermembrane, okay, from matrix into intermembrane space, from intermembrane space back into the matrix through ATP synthase, and ATP synthase will synthesize ATP molecule. So yang ni saya just tunjuk the whole diagram. Okay, so the 4 stages of cellular respiration: glycolysis in the cytosol, breaks glucose, okay, into pyruvate. 1 glucose becomes 2 pyruvate. So the reaction produces a net of 2 ATP through substrate level phosphorylation. So the process also occurs and produce NADH. So NADH, you have to take note, is produced in the cytosol. Okay, so how many NADH is produced from glycolysis stage? Also 2. Okay, also 2 molecule. So but for the NADH molecule that is produced during glycolysis, it maintains inside the cytosol and it cannot enter into the mitochondria. So this NADH cannot enter mitochondria. So what happened is that the electron of NADH will be passed using electron shuttle. Electron shuttle dekat membrane ni akan ada electron shuttle. Okay, shuttle. So the electron of NADH will pass to electron shuttle in the mitochondria. So this electron shuttle will pass the electron to either NAD plus or FAD. So it depends on cell type. Okay, so if we look at liver, so the acceptor will be NAD plus. If it's involved the brain cell, the electron acceptor will be FAD. So for brain. So ini awak kena take note sikit. Okay. So the NADH produce the glycolysis cannot enter into the mitochondria, but it passes the electrons, its electron to electron shuttle, and then electron shuttle to either NAD plus or FAD. Depends on the cell type. Okay. So the next stage is pyruvate oxidation. Okay, so pyruvate oxidation. So convert acetyl-CoA. Convert pyruvate into acetyl-CoA, and then this stage, okay, also produced NADH. But the NADH produced is already inside the matrix. So itu tak ada masalah. Okay, it is already inside the mitochondrial matrix. So the next stage is the citric acid cycle. So citric acid cycle also produces NADH and also FADH, 2. Okay, so and it also produce ATP molecule. Yang ini for citric acid cycle, you have to know how many ATP molecule is produced. Satu awak kena tahu, for one complete cycle, it only produces 1 ATP. But for 1 molecule of glucose, okay, for the whole reaction, for 1 molecule of glucose, it requires the citric acid cycle to occur 2 times. Okay, citric acid cycle will occur 2 times. Okay, so it means that for citric acid cycle, it produces 2 ATP molecule through substrate level phosphorylation for 1 molecule of glucose that the reaction consumes. Okay. And then the last one is oxidative phosphorylation. So for oxidative phosphorylation to occur, it requires these 2 molecules to enter into the phase. Okay, so this will act as electron carrier and donates the electron to the electrons carriers that are embedded in the crystal. And then the electron will be transferred from one electron carrier to another one, and releases energy. So this energy is used during chemiosmosis to move proton across the membrane of mitochondria, the inner membrane, and produces ATP molecule. Okay. So the amount of ATP produced, okay, for oxidative phosphorylation is different, depends on the type of cell. Okay, because you have to take note for these facts. The NADH that is produced during the glycolysis stage. Okay, so if it's liver cell, the amount produced is 32. If it's brain cell, the amount is 30. So nanti kita tengok kenapa ada perbezaan ni. Kenapa boleh lead kepada 30 dengan 32 ni. Okay. So glycolysis. As you can see from the name, okay, lysis it means to break. Lysis, it breaks. Breaks what? Carbohydrates, glyco. Okay, so that's why it is called as sugar splitting. Okay, so break glucose into 2 molecules of pyruvate. Glucose 6 carbon molecule. Pyruvate is a 3 carbon molecule. Okay, so it produces 2 pyruvate at the end of the reaction. Occurs in the cytoplasm. 2 phases: energy investment phase and splitting of sugar. So in this stage, 2 ATP are used. And then the next phase is energy capture phase, or another term is energy payoff phase, that produces 4 ATP. So that's why I said before, you have for glycolysis, you have to take account the net production of ATP only. Okay, so maksudnya berapa yang terhasil? 4. Okay. Tolak dengan 2 yang dah digunakan. So maksudnya the net production is 2 ATP for glycolysis. Okay. So this glucose ni dia nak menggambarkan apa? Glucose 6 carbon, pyruvate is a 3 carbon sugar. Okay. So the energy input and output of glycolysis. So for energy investment phase, glycolysis ni dia ada 10 reactions. Okay. Energy investment phase daripada reaction yang pertama sampai reaction yang kelima. Energy payoff phase, reaction yang ke-6 sampai reaction yang ke-10. So for the first phase, 2 ATP is used. Okay. And when ATP is used, means that the ATP is hydrolyzed to release energy, and also the product of the breaking of ATP is ADP and also 2-phosphate. And then for energy payoff phase, 4 ATP produced, 2 NADH produced, and 2 pyruvate produced. So the net production is 2 pyruvate, 2 ATP, and also 2 NADH. Yang ini awak kena ingat. Okay. Glycolysis also produces water. Okay. So this is the whole reaction, the whole 5 reaction, the first 5 reactions of the first phase, which is energy investment phase. Reaction 1, reaction 2, 3, 4, and 5. Okay, so in the initial reaction involved glucose to enter, and then at the end of phase 5 ni, the fifth reaction, it produces basically 2 molecule of G3P molecule. Okay, that enters into reaction 6 in the second phase. Okay, so kalau tengok dekat dalam reaction ni, reaction yang pertama dengan reaction yang ketiga akan menggunakan satu ATP each. one ATP molecule each. Kalau tengok kat glucose kan, glucose initially it does not have any phosphate attached to it. So when ATP enters, so ATP releases one of its phosphate to the glucose molecule, so glucose become glucose 6-phosphate. Okay, nampak dia akan attach pada carbon number 6. So that's why we call it as glucose 6-phosphate. Carbon number 1, 2, 3, 4, 5, 6. So it is called as glucose 6-phosphate. And then comes an enzyme which is phosphoglucoisomerase. Daripada nama isomerase, maksudnya dia akan convert glucose into its isomer, which is fructose. Okay, so now it becomes fructose 6-phosphate. Okay, sebelum kita proceed ke reaction yang ketiga, tengok nama enzim ni kinase. Mana-mana enzim yang ada nama kinase, in this case hexokinase, okay, so this enzyme involves the transfer of phosphate group. Okay, the transfer of phosphate group. So same goes to reaction 3. Okay, so phosphofructokinase. Okay, so maksudnya bila ATP masuk, ATP akan release phosphate dia kepada fructose 6-phosphate. Okay, nampak? Carbon yang pertama, kedua, ketiga, keempat, 5, 6. So that's why it is called fructose 6-phosphate. Okay, so ATP masuk, release phosphate dia kepada molekul yang ni. Okay, so dia akan jadi fructose 1,6-biphosphate. Okay, fructose 1,6-biphosphate, bisphosphate. So phosphate akan attach pada carbon number 1 and carbon number 6. Okay, fructose 1,6-2-phosphate, bisphosphate. Okay. So the next one is aldolase. So aldolase will convert fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate and another molecule which is dihydroxyacetone phosphate, DHAP. So both of these molecules are 3 carbon sugars. But in order to proceed to reaction 6, you don't want DHAP, tak nak. You want to convert DHAP into G3P using isomerase. Okay, so only G3P can proceed into the next reaction, which is reaction 6 in the second phase of glycolysis. Okay, so so far soalan tak pernah lah tanya. Okay, tak pernah tanya soalan exam. Soalan exam tak pernah tanya okay, name the enzyme that convert glucose to glucose 6-phosphate. Tak ada pernah tanya. Okay, cuma awak kena tahu bila masa ATP masuk, apa fungsi ATP. Kalau awak tengok nama enzyme ni kan, isomerase, what does it do kan? To convert glucose into fructose. Okay, so isomerase convert DHAP to G3P. So you have to know all this. Okay, so kalau macam first phase dia akan produce, dia akan used to ATP molecule. Macam itulah dia punya initial substrate dia adalah glucose. Akhir dia akan hasilkan 2 G3P. Macam tu je. Okay. So the next slide it explains the detail of the reactions reaction 1 until reaction 5. So saya dah explain dah, so I just skip. So the next phase of glycolysis adalah energy capture phase or energy payoff phase. Dia bergantung pada buku. Kalau buku rujukan awak adalah Solomon kan yang kena beli hari tu, so dia akan guna istilah capture phase. Tapi kalau ada siapa-siapa yang buku senior, buku Campbell, dia guna istilah payoff phase. So mana-mana pun okey, benda yang sama. Okay, so kalau awak tengok kat sini, the second phase, energy capture phase, adalah reaction yang ke-6 sampai yang ke-10. Okay, so in this phase, awak kena dah start kalikan all the intermediate substrate kan and the products dengan 2. Sebab apa? Yang akan masuk dekat reaction ke-6 ni adalah 2 molecule of G3P. Okay, 2 molecules of G3P molecule. So nampak kalau kat sini, nampak kat sini kan, dia dah kalikan dengan 2 siap-siap semua 2. Sebab apa? Sebab yang akan masuk 2 molecules of G3P. Okay, so G3P, glyceraldehyde 3-phosphate. Molekul ni dekat carbon yang ketiga ada fosfate. So that's why kita namakan dia sebagai glyceraldehyde 3-phosphate. And then comes the enzyme dehydrogenase. Tadi saya explain kan, dehydrogenase removes atom from a substrate and gives to another substrate. So in this case, the hydrogenase enzyme removes hydrogen atom from G3P and give it to NAD plus to become NADH. Okay, so releases hydrogen ion also to the surrounding. So ingat semua kena kalikan dengan 2. Lepas tu dia akan jadi apa? Ni jadi reduced lah. So NADH is produced from the reduction of NAD plus. Okay. And then come 2 inorganic phosphate added to the substrate. So become 1,3-bisphosphoglycerate. Okay. So for this molecule, if you look at this molecule compared to G3P, okay. So nampak asalnya memang dah ada fosfate kat carbon number 3. Cuma bila fosfate datang kat sini, dia akan attach kat number 1. So that's why the molecule is called as 1,1,3-bisphosphoglycerate. So molecule ni ada 2 fosfate attached to it. Okay, so mana-mana molecule yang ada dua phosphate attached to it, it is a highly energized molecule. Okay, and highly energized molecule. So it has to release one of its phosphate to ADP. Okay, so the reaction involved, if you can see from the diagram here, the enzyme kinase. So kinase akan remove phosphate yang dekat carbon pertama, pergi dekat ADP, so ADP becomes ATP. So now the product produced is 3-phosphoglycerate. Okay, 3-phosphoglycerate and then converted into 2-phosphoglycerate kan, sebab dia just tukarkan je position phosphate tu daripada carbon nombor 3 kepada carbon number 2. Okay, dia just convert kan. So phosphogluco, so guna enzyme phosphoglyceromutase. Okay, so and then convert to PEP with the release of water, and then PEP convert into pyruvate. So the reaction also produces ATP. So nampak PEP ni phosphoenolpyruvate ada phosphate. So phosphate is released to ADP, ADP becomes ATP, and then at the end you produce pyruvate. 2 molecules of pyruvate is produced. Okay, will be produced. Okay, so from 1 glucose, okay, undergo the 2 phases of glycolysis, produces 2 pyruvate. Okay, 2 pyruvate. So these are the details of glycolysis. Okay, itu je lah kan. So kita take a break for 2 minutes or digestkan apa yang saya dah cerita tadi daripada mula sampai yang glycolysis. Kita rehat sekejap. Ingat ya, dia kalau peringkat awal ni awak tak payah nak hafal nama-nama enzim tu kan. Intermediate product, so far tak pernah tanya, cuma kena tahulah flow of the reaction, the flow of the reaction.
Meja.
Ya.
Kena ingat lukisan 1,3-bisphosphate cellulate apa tu?
Structure tak payah ingat. Cuma saya explain tadi supaya tahu kenapa lah nama dia macam ni pelik kan. Ada 2, ada 1, ada 3. Sebab attachment of phosphate tu je. Okay?
Thank you, madam.
Okay. Okay, so we are going to continue on the second phase of glycolysis, which is the pyruvate oxidation. So this is the second phase and this is the third phase. Okay, so both phases occurs in the matrix of mitochondria. Okay. Okay, kita tengok yang phase yang kedua ni, okay, which is pyruvate oxidation ataupun nama dia link reaction ataupun formation of acetyl-CoA. Okay, so why do we call it as pyruvate oxidation? Because pyruvate is oxidized. Okay, how is it oxidized? Because in the reaction, it will reduce the it will cause NAD plus to be reduced to form NADH. Okay, so in this reaction, pyruvate enters mitochondria via active transport. It is also called as link reaction, which is it links between glycolysis and also citric acid cycle. Kenapa pyruvate ni kena ditukarkan kepada acetyl-CoA? Kenapa dia tak boleh masuk je kan? Sebab glycolysis it occurs in the cytosol. Okay, pyruvate yang terhasil ni awak tengok dia punya molekul. Molekul dia ada charge. Okay, the molecule it has charge. It has the carboxyl group here that has charge, COO negative. Kita dah belajar dekat chapter 3, awak tahu feature of membrane. Membrane apa feature dia? Membrane dia tak boleh, it cannot allow charge molecule to pass through or ion to pass through. So in order to allow pyruvate to enter, it has to be transported using this transport protein. Dia tak boleh, this pyruvate molecule cannot just easily diffuse across the membranes of mitochondria. Membranes of mitochondria, it has two membrane kan, outer and also inner membrane. So pyruvate just cannot simply diffuse through the membrane of mitochondria. It has to be transported into the matrix using this transport protein. Okay. So once pyruvate enters through this transport protein, it will be oxidized. Okay, oxidized. And when it is oxidized, it will lose its oxygen atoms here. Okay. So this part of the pyruvate will be released as carbon dioxide. So that's why dicakap kat sini pyruvate is oxidized. Okay, because oxygen here will be released and removed as carbon dioxide. And in the process, okay, of the reaction, NAD plus will be reduced to NADH plus H plus. Okay, so pyruvate now, because it has removed its carboxyl group here, one carbon is removed and now it becomes a 2-carbon compound. We call it as acetate. Okay, acetate, this region here is acetate. So acetate that enters will combine with coenzyme A, become acetyl-CoA. Okay, so if you look at this diagram here, the link or the bond between acetate and the coenzyme A is through a very unstable bond. Okay, it is a very unstable bond. So the attachment is just only temporary. Okay. So those are the 3 reaction lah. So first bila dia masuk, dia akan release carbon dioxide. First yang pertama oxidized. And second, it causes NAD plus to be reduced to NADH. And third, coenzyme will come in and then attach to the acetate. Kan ini akan jadi komponen yang kita panggil dia sebagai acetate. And then attach to CoA and then become acetyl-CoA. So those are the 3 reactions of pyruvate oxidation. Okay, so pyruvate oxidation ada 3 reaction yang berlaku. Carbon dioxide released, NADH produced, and acetyl-CoA produced. Three reactions, okay, in this stage, in the link reaction stage. Okay, so the next stage is citric acid cycle. So another name for citric acid cycle is 3-carboxylic acid or Krebs cycle. So ini kita jarang guna lah, so just ignore ini. either this one. Okay, tapi ini yang standard yang this one here. Ni nama lama dia. Okay, yang ni yang sepatutnya awak guna, citric acid cycle. Okay, so it takes place in the mitochondria. It was discovered by this scientist. Okay. And then, so this is the structure of mitochondria. So, Krebs cycle or citric acid cycle, it occurs in the matrix of mitochondria. So, why does it occurs in the matrix of mitochondria? Because this reaction involves enzyme that are found in the matrix of mitochondria. Okay, and it also releases its product, which is NADH and also FADH. Two, which are also released in the matrix of mitochondria. Okay. So as you can see from the diagram, the inner membrane here, the blue color is the inner membrane. Sorry, the yellow color structure here is the inner membrane or cristae. So the cristae has many electron carriers of the electron transport chains, okay, of the electron transport chains that are embedded in the cristae. And what else can you find in the cristae? The ATP synthase. Okay, the ATP synthase also embedded in the cristae. ATP synthase, it has two functions. One as channel protein that involves in the movement or diffusion of hydrogen ions. And second, it functions as enzyme, okay, to synthesize ATP. Okay. So in the matrix, you also have ribosome lah, kan? So the function of ribosome is to produce all these proteins, okay, in the mitochondria, especially in the matrix of mitochondria. Okay, so for the citric acid cycle, it has 8 steps or 8 reactions. Reaction 1, 2, 3, 4, 5, 6, 7, 8. Okay, so for one cycle, for one complete cycle, it produces 1 ATP, 3 NADH, 1 FADH2. This is for one cycle that requires 1 acetyl-CoA. Tapi awak kena ingat, step yang sebelumnya adalah glycolysis, kan? Glycolysis yang menggunakan satu molekul glukos. Okay, so glukos ni akan break down into 2 pyruvate dan akan menghasilkan 2 acetyl-CoA. Okay, so for 1 glucose, the product that is produced for citric acid cycle, awak kena kalikan dengan 2. So ini sebenarnya untuk for the production of this product, for 1 glucose, you have to times by 2. So for 1 molecule of glucose akan menghasilkan 2 ATP, 6 NADH dengan 2 FADH2 molecule for 1 glucose, okay, through citric acid cycle. Citric acid cycles, okay. So kalau tengok from this diagram, so acetyl-CoA is a 2 carbon compound. When it enters into reaction 1, it will combine to this compound, which is oxaloacetate. Oxaloacetate is a 4 carbon compound, okay. So acetyl-CoA bila dia masuk, kan tadi saya cakap kan, ikatan antara acetate dengan CoA ni adalah sangat tak stabil. So CoA akan terus released. Okay, and once it enters, it is released from the acetate. So acetate combined with oxaloacetate becomes citrate. Okay, so citrate is a 6 carbon compound. Okay, nampak? 4 campur 2 jadi 6. So it produces citrate is a 6 carbon compound, and then water leaves and then re-enters to just rearrange the atoms within the molecule. So citrate become isocitrate, rearrangement. Okay, so reaction yang ketiga is the production of NADH. So NAD plus is reduced, produces NADH, and in reaction 3 also carbon dioxide is released. Okay, so citrate and isocitrate is a 6 carbon compound. Bila satu carbon dioxide keluar, so maksudnya produk yang terhasil dekat reaction yang daripada reaction yang ketiga ni is a 5 carbon compound. Okay. Alpha ketoglutarate is a 5 carbon compound sebab kat sini dah release carbon dioxide. Okay. So alpha ketoglutarate, a 5 carbon compound. So the next reaction, reaction 4 will release another carbon dioxide. So dah kekurangan lagi satu carbon dioxide kan. So maksudnya produk yang terhasil dekat sini is a 4 carbon compound. So daripada alpha ketoglutarate nak jadi succinyl-CoA akan release carbon dioxide dan akan menghasilkan NADH. Okay. So ini CoA masuk balik kan, so jadi succinyl-CoA. Yang ni stage yang sangat penting awak kena ingat. Stage yang kelima, from succinyl-CoA it is converted into succinate and in reaction 5 ATP is produced. Kalau awak tengok kat sini, succinyl-CoA ni dia akan apa yang akan berlaku, inorganic phosphate will enter into the reaction. Maksudnya inorganic phosphate mula dia akan attach kat molekul ni, tapi lepas tu dia akan bagi kat GDP. GDP is a molecule, we call it as guanosine diphosphate. GDP sama juga dengan ADP, cuma dia punya base dia berbeza. ADP adenosine diphosphate, ni guanosine diphosphate. Base dia je yang berbeza. Okay. So ADP campur dengan inorganic phosphate jadi GTP. Okay. Guanosine triphosphate. So fosfat daripada GTP ni dia bagi dekat ADP jadi ATP. Okay. So yang ini adalah product of citric acid cycle. Okay. For one complete cycle. So succinyl-CoA, as I mentioned kan, CoA ni adalah sangat tak stable dalam ikatan tu, so dia akan release. Dia masuk sekejap je, masuk keluar. Okay. So succinate akan convert kepada fumarate. So in the reaction, it reduces FAD to become FADH2. Okay. So FAD becomes FADH2, fumarate also. Ini the start daripada sini, semua dah jadi 4 carbon compound. 4 carbon compound. So fumarate convert into malate, water is added. So water from the malate will be the hydrogen atom will be given to NAD plus, become NADH. Okay. So nampak dalam reaction citric acid cycle ada 3 NADH hasil. 1, 2, 3. This one here. ATP 1. This one here. FADH2 terhasil dekat reaction nombor 6. So 1 here. Okay. So hasilkan oxaloacetate balik semula. So it is a cycle. Okay, the process is a cycle. Okay. So these are the detailed reactions. Saya dah explain. So as a summary for citric acid cycle, awak kena tengok balik daripada proses glycolysis. So glycolysis uses one molecule of glucose that breaks into 2 pyruvate. Okay, 2 pyruvate. And then from 2 pyruvate undergo link reaction or pyruvate oxidation to produce acetyl-CoA. So setiap satu ni akan masuk dekat citric acid cycle. So setiap cycle akan menghasilkan 3 NADH, 1 FADH2, 1 ATP. So untuk satu glukos, total daripada awal glycolysis, daripada link reaction and citric acid cycle ni akan menghasilkan a total of 8 NADH. 1, 2, campur 3 kat sini, campur 3 kat sini, 6 kan? 6 campur 2, 8. So 2 FADH and 2 ATP. Kenapa total output ni dia tak kira product of glycolysis juga? Sebab kan tadi saya cakap glycolysis ni berlaku dekat dalam cytoplasm. NADH dia terhasil dekat dalam cytoplasm, which bila dia masuk kat dalam mitochondria, dia tengok sel. Sama ada brain cell ataupun liver cell. Kalau brain cell, NADH akan jadi still NADH. FADH dia akan tukar jadi, NADH akan tukar jadi FADH. Kenapa dua molekul ni menghasilkan jumlah ATP yang berbeza? Kalau untuk satu molekul NADH, bila dia masuk oxidative phosphorylation nanti, dia akan menghasilkan 2.5 molekul of ATP. Tapi kalau FADH yang masuk dalam oxidative phosphorylation, dia akan hanya menghasilkan 1.5 ATP sahaja. So, awak kenalah kalikan nanti, awak kena tambah kalikan semua kalau macam kat sini kan. Contohnya kalau dia merupakan liver cell. Okay. So, maksudnya awak kena tambah kat sini ada lagi 2 NADH. So, kalau dia merupakan liver cell, total berapa? 10 NADH kan? Kali dengan 2.5. Dah berapa? 25 ATP yang terhasil daripada NADH dalam liver cell. Campur dengan 2 FADH. 2 FADH kali dengan 1.5 dapat berapa? Dapat 3. Okay. Campur dengan 2 ATP kat sini. Kan? 2 ATP. Campur kat sini glycolysis awak kena ingat. Net untuk glycolysis ada 2 lagi ATP. Campur lagi 2. Tak muat dah. Campur lagi 2. So dah berapa? 25. Ni untuk liver 25 kat sini. Campur dengan 3 ATP. Campur dengan 2 yang ni. Kan? Campur dengan 2 ATP kat sini masa glycolysis. Berapa semua? Ini 25 campur 3. 28, 29, 30, 31, 32. 32 ATP untuk liver. Macam tu. Okay, so nanti kita tengok detail calculation dia. Ini awak kena tahu macam mana calculate ATP untuk kedua-dua sel ni. Okay. Okay. Kita break 2 minit jap. Digest dulu benda ni. Kenapa dia jadi macam ni? Dekat awak punya slide. Cuba try dekat dalam kertas kosong, awak kirakan untuk liver cell dengan brain cell, awak asingkan produk dia. Glycolysis, link reaction, Krebs cycle, ada berapa NADH, FADH dengan ATP yang terhasil dalam liver dan juga dalam brain. Ingat brain, NADH dia akan bertukar jadi FADH. 2. Sorry FADH2. Cuba ambil satu kertas kosong, cuba scribble. Jumlah output untuk glycolysis, link reaction, citric acid cycle untuk brain dengan untuk liver. Dan tulis kat satu corner, 1 NADH sama dengan 2 ATP yang akan terhasil. 1 FADH2 akan menghasilkan 1.5 ATP. Okay, so kita nak masuk sekarang dekat the last phase. Okay, the fourth phase of oxidative phosphorylation. So this stage involves the electron transport chain and also chemios. Ni kat sini, chemiosmosis. Okay, chemiosmosis. So kalau tengok kat diagram ni, if you look at the diagram, so for the first component ETC, they are actually composed of several electron carriers. So these are electron carriers: complex 1, complex 2, complex 3, complex 4. Okay, so these are electron carriers. So they are electron carriers, but where is the source of electrons? The source of electrons come from the NADH and also FADH2 that was produced during glycolysis, link reaction, and also citric acid cycle. Okay, remember glycolysis produce 2 NADH. Link reaction also produce 2 NADH. Citric acid cycle produces for one complete cycle is 3. Tapi kita nak consider for one molecule of glucose. So maksudnya dia akan hasilkan 6 NADH and also 2 FADH molecule. So these are the products that will enter into the ETC or oxidative phosphorylation. But for this NADH, you have to take note that it is different in different cell. So for liver, it still becomes NADH, but for brain, it becomes FADH2. Okay, so kalau kita bahagikan kat sini, liver, brain. Okay, so jumlah NADH ada berapa? Kita cuba kira ya. Kita cuba kira. Kalau untuk liver, kat sini ada 2 NADH, kat sini ada 2 FADH. Okay, liver 2 NADH masa glycolysis, 2 masa link reaction, 6 masa Krebs cycle. So jumlah 10 NADH untuk liver campur dengan 2 FADH2. Ini awak kena ingat. Okay, liver. Kalau brain, instead of 2 NADH, for glycolysis, dia akan convert jadi 2 FADH2, 2 NADH untuk link reaction. Yang atas glycolysis, link reaction, Krebs cycle still produce 6 NADH dengan 2 FADH2. Okay, so ini adalah produk untuk brain. So brain maksudnya dia akan hasilkan 8 NADH dengan 4 FADH2. So kalikan je lah kan yang tadi tu kalau dia punya value. Kalau apa? NADH sama dengan 2.5 ATP, FADH2 sama dengan 1.5 ATP. Kalikan berapa semua ATP yang terhasil semasa oxidative phosphorylation. Campur dengan berapa banyak ATP yang terhasil semasa substrate level phosphorylation yang berlaku semasa glycolysis dan juga semasa Krebs cycle. Okay, semasa Krebs cycle. So yang ini awak kena ingat, ni adalah antara soalan yang memang suka keluar lah dalam exam. Okay. Okay, so oxidative phosphorylation. Okay, so we are going to look at first the component of electron transport chain of oxidative phosphorylation. So as I've mentioned, it has four protein complexes. Okay, so complex 1, complex 2, complex 3, 4. Okay, so for complex 1, they also function, for complex 1, kalau tengok kat sini, complex 1, 3 and 4, these are protein that are embedded in the mitochondrial membrane. Okay, in the mitochondrial membrane. So they are actually integral protein. Ini chapter berapa? Chapter 3, ingat Integral proteins are proteins that are embedded in the phospholipid bilayer. So this protein complex, apart from it has role as electron carrier. Another function of this protein complex 1, 3 and 4, they also function as proton pump. Okay, ingat bila awak jumpa perkataan pump, maksudnya dia merupakan protein carrier, transport protein yang terlibat dalam aktif transport. Ingat. Proton pump, proton maksudnya hydrogen ion. So dia melibatkan transport protein yang melibatkan the transportation of hydrogen ion through active transport. It means that the movement of hydrogen ion is from low concentration to high concentration of hydrogen ion. So that is the function of proton pump here. It pumps hydrogen ion from low concentration to high concentration. So if you look at this diagram, the yellow region here is the matrix. Okay, matrix of mitochondria. The orange region here is the intermembrane space. Okay, it is the intermembrane space. So, mana nak datang? Kalau kita belajar kan, active transport ni, dia memerlukan energi. Mana datang energi ni? Energi ni datangnya daripada the movement of electrons from one carrier to another carrier to another carrier, and the electrons is provided by the NADH molecule or FADH molecule. So NADH ataupun FADH molecule, FADH2 molecule will provide highly energized electrons. Okay, so NADH akan riliskan dia punya hydrogen ion, nampak? Bila dia masuk dalam reaction, NADH ni will become oxidized. Okay, oxidized, release its electron and hydrogen ions. Hydrogen ions akan release into the matrix. Okay, so these hydrogen ions, okay, will be pumped from the matrix into the intermembrane space from low concentration to hydrogen ion region that has high concentration region. Okay, so from low concentration to high concentration across the proton pump. Okay, across the proton pump here. So they can pump across. So hydrogen ion will be accumulated in the intermembrane space. Hydrogen ion will be accumulated in the intermembrane space. So once hydrogen ion is accumulated, you have a high concentration of hydrogen ion in the intermembrane space. What happens next is that the hydrogen ion will diffuse back into the matrix, okay, using this protein, ATP synthase. Okay, so ATP synthase, dia ada dua fungsi macam saya cakap tadi. Satu sebagai protein channel. Ingat kalau channel, dia terlibat dalam passive transport, which is facilitated diffusion. Okay, facilitated diffusion. Satu lagi fungsi dia sebagai enzyme. So enzyme untuk synthesize ATP. Okay. So as hydrogen ion concentration is high in the matrix, it diffuse back into the as hydrogen ion is high in the intermembrane space, it diffuse back into the matrix using ATP synthase, and the ATP synthase will synthesize ATP from ADP and also inorganic phosphate. So this is the chemiosmosis process of oxidative phosphorylation. Okay, so ini merupakan facilitated diffusion lah. So macam mana ATP synthase ni hasilkan ATP ni? Dia sebenarnya akan menggunakan satu bentuk energy from the diffusion of hydrogen ion. We call it as proton motive force. Proton motive force, which is the high concentration of hydrogen ion from the intermembrane space that diffuse across the membrane into the matrix. Itu adalah proton motive force. Ataupun hydrogen ion electrochemical gradient. Okay, hydrogen ion electrochemical gradient. Okay, so kalau awak tengok yang kat sini pula, kita ada the second complex. Okay, which is a peripheral protein. Nampak? Maksudnya it is not embedded in the crystine. So yang electron carrier yang lain adalah Q. Q stand for ubiquinone, mobile carrier. It is not attached to anything. And then it can move. And then you have another one which is cytochrome C, also a mobile carrier. Kalau awak tengok tadi kan, saya mention satu NADH akan menghasilkan 2.5 ATP, satu FADH2 akan menghasilkan 1.5 ATP. Kenapa ada berlaku perbezaan penghasilan ATP? Kenapa NADH lagi banyak berbanding dengan FADH2? Kenapa NADH 2.5, FADH2 1.5? Sebab awak kena tengok dekat diagram ni. NADH dia masuk lebih awal dalam ETC tu. Dia masuk daripada kompleks 1. Tapi kalau FADH2 dia masuk dekat kompleks yang kedua. Maksudnya bila NADH dia masuk lebih awal, maksudnya dia akan release lebih banyak energi kepada ETC tu. Maksudnya lebih banyak proton akan dipump masuk ke dalam intermembrane space berbanding kalau FADH2 masuk lambat sikit kan. Dia masuk dekat kompleks yang kedua. So dia akan release less energy from the electron movement. So electron akan hydrogen ion hanya akan pump daripada yang kompleks 3 ni. Okay, so the energy release from FADH2 is lesser compared to NADH molecule. So that's why jumlah ATP untuk NADH ni lebih banyak berbanding dengan FADH2. Dia masuk, FADH2 masuk lambat, NADH masuk cepat. Banyak lagi energi yang terhasil untuk pump hydrogen ion daripada matrix ke dalam intermembrane space. So itu je sebab perbezaan jumlah ATP yang terhasil tu. Okay, so next, the electron transport chain. Saya dah mention tadi, a collection of protein complexes and organic molecules embedded in the crystalline. Okay, dia ada 4 kompleks. Kompleks 1, kompleks 2, kompleks 3 and 4. 1, NADH akan masuk. 2, FADH2 akan masuk. Okay, and if you look at the diagram also, here, tengok kan. Kat sini, okay. The last electron acceptor will be oxygen molecule. So oxygen molecule akan combine dengan hydrogen ion in the matrix, okay, and also it receives the electron and it becomes water, okay. So water, as you know, is the byproduct of cellular respiration. Okay, so this is another diagram to portray you how the electrons are transported from one electron carrier to another electron carrier. So kalau tengok kat sini, Dia ada 4 kompleks. Kompleks 1, kompleks 2, 3, 4. Dan dia kalau tengok kat sini merupakan macam satu graf. Okay. So dia kalau tengok kat sini dia elektron ni asalnya it is highly energized. Okay. So when the electrons are released from the NADH molecule that function as the electron carrier, so the electron is released and pass from one carrier to another carrier to another carrier to another carrier and then the next one. Tengok dia semakin ke bawah. Sebab apa? Sebab as the electron are passed from one carrier to another carrier, it releases energy. So this energy is used during chemiosmosis to produce ATP molecule. Okay. So lastly, when the energy is transferred from one carrier to another carrier and it is received by the last electron acceptor, okay, which is oxygen molecule. Ingat oxygen molecule is the last electron acceptor. Bila dia bergabung dengan elektron, dia akan juga bergabung dengan hydrogen ion in the matrix dan dia akan jadi air. Okay, so kalau tengok kat sini, electrons from either NADH and also FADH2 move from a less electronegative electron carrier to a more electronegative electron carrier down the chain and releases energy. Maksudnya semakin ke bawah electron carrier tu, semakin dia lebih electronegative. Sebab apa dia nak? The function of the electron carrier is to pull electron. Okay, it is to pull electron towards it to receive the electron and then pass to a more electronegative electron carrier. So maksudnya kat sini, oxygen is the most electronegative electron carrier. It is the most electronegative electron carrier. Sebab apa? Dia fungsi untuk tarik elektron tu sampai didapat elektron tu. Okay, so dia melibatkan proses oxidation and reduction reaction. Receive and then donate. Receive and then donate. So ini pun dah mention kan. So NADH, point 1, NADH and also FADH2 donate electrons to the electron transport chain. Okay, second, electrons are passed from one electron carrier to another electron carrier. So it involves redox reaction, receive and then donate, receive and then donate, oxidize, reduce, and then the process release energy. Energy comes from the electron movement, okay, the electron transfer. And then the energy released from the electron, okay, is used to pump hydrogen ion. Pump, ingat the word pump is active transport. So from matrix into intermembrane space. Okay. Hydrogen ion concentration is low in the matrix. Hydrogen ion concentration is high in the intermembrane space. Pump, okay, by proton pump. So these are proton pump. Okay, the electron carrier also function as the electron pump. So from matrix into intermembrane space, electron drop in free energy. Maksudnya as it pass from one carrier to another carrier, the electron releases energy, okay, for chemiosmosis to occur to produce ATP molecule. Okay. So chemiosmosis of producing ATP using energy in the proton electrochemical gradient. Ni lah kan perbezaan hydrogen ion concentration. Okay. High in the intermembrane space and low in the matrix. So hydrogen ion diffuse down. So melalui dia akan menghasilkan proton motive force. Okay. The movement, okay, of the hydrogen ion provides proton motive force for the ATP synthase to synthesize ATP. Okay, so kita tengok chemiosmosis is the energy coupling mechanism. Chemiosmosis is process of producing ATP using energy in hydrogen ion electrochemical gradient. Okay, high concentration of hydrogen ion in the intermembrane space. Okay, so the proton gradient or the proton motive force creates or has energy. Okay, it has energy. The high concentration of hydrogen ion in the intermembrane space will diffuse back into matrix. Sebab apa? Dia menghasilkan tenaga ni. Proton motive force. Ini adalah sebentuk tenaga. Okay. So yang digunakan oleh ATP synthase untuk synthesize ATP. So ATP synthase it is embedded in the inner membrane of mitochondria ataupun piste. Okay, so hydrogen ion diffuses back into the matrix through the ATP synthase, causing ATP synthase to synthesize ATP. It phosphorylate ADP and becomes ATP. Okay. Istilah ni awak kena ingat ni. Dia menghasilkan proton motive force ni. So it says here electron transport and pumping of proton, which creates an a proton gradient across the membrane. Okay. So from low to high and then back high to low. Active transport in this part, passive transport in this part. Okay. So this is the structure of this is how ATP synthase looks like. Okay, so daripada high concentration of hydrogen ion in the intermembrane space, dia akan masuk ke dalam ATP synthase ni, and the hydrogen ion will causes the rotor part of the ATP synthase to rotate. Okay, so the rotation causes the next portion of the ATP synthase, which is we call it as the catalytic knob. Okay. To phosphorylate ADP and also inorganic phosphate to become ATP. Okay. Kita break sekejap sebelum kita masuk kepada calculation. S11 ada kelas pukul 11. Sorry, S10 ada kelas pukul 11
Ada madam.
Ha?
Ada madam.
Ada. So you all boleh leave. Saya sambung dengan S9. S9
Tak ada madam. Tak ada madam.
so saya sambung dengan S9. Nanti
Tak ada madam.
recording tu saya bagi dekat S10 untuk tengok balik semula ya.
Thank you, madam.
Okay.
Thank you madam.
Okay.
Madam.
Okay.
Madam.
Thank you, madam. Okay.
Thank you, madam.
Okay. Okay. Kena sambung untuk S9 on calculation of ATP. Tinggal sikit je lagi ni. Okay. So ATP calculation from cellular respiration. So you have to know the products of glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation. Okay, so how much number of ATP produced after oxidation of one molecule of glucose? So I always emphasize for you to know what is the products of each of the stage for one molecule of glucose, but not for one molecule of acetyl-CoA. Okay, for one cycle of citric acid cycle. One cycle of citric acid cycle only accounts for half of a glucose. Okay, so for one molecule of glucose, it requires the citric acid cycle to occur 2 times. It requires the product of pyruvate oxidation to acetyl-CoA, 2 molecule of acetyl-CoA. So kalau kat sini kan, if you look at in the diagram, okay, glycolysis, it produces 1 molecule of NADH and 2 molecule of ATP. The 2 molecule of ATP is through substrate level phosphorylation. Citric acid cycle also produce 2 molecule of ATP through substrate level phosphorylation. The amount is really small compared to the amount of ATP produced during oxidative phosphorylation, because for oxidative phosphorylation, it uses the power of electrons from NADH molecule and also FADH2 molecule that was produced from the previous stages of of cellular respiration, which are glycolysis, pyruvate, oxidation, and also citric acid cycle. So oxidative phosphorylation will produce the most ATP molecule. So about 26 or 28 ATP molecule. So it depends on the cell type of cell. So for brain, for example, it produces 26. For liver cell, it produces 28. Okay. So kalau kat sini, awak dividekan dia kepada beberapa kolom. Okay. This one are stages of cellular respiration. Ini brain. Ini liver. Okay. Kita tengok stage yang pertama adalah glycolysis. Glycolysis, dia menghasilkan 2 molekul NADH dekat dalam cytoplasm. Saya cakap tadi, 2 molekul cytoplasm ni dia tak boleh masuk dalam mitokondria. So, apa yang terjadi adalah elektron daripada NADH ni akan diberi kepada elektron shuttle yang ada pada inner membrane of mitochondria. Okay, so electron of NADH akan bagi kepada sama ada NAD plus ataupun FAD saja yang terletak dalam matrix of mitochondria of either of the two different type of cell. So kalau NAD plus, dia elektron tu diterima dalam liver cell. Ini dalam brain cell. So maksudnya kat sini untuk brain, instead of 2 NADH, saya kena pastikan kolom lagi ni. Okay. Produk dia adalah NADH. Okay. Untuk brain akan terhasil NADH tak? Tak ada. Okay. Liver ada tak? Ada. Berapa? 2. Okay. So second stage. Sekejap. Padam lagi. Sekejap ya. Saya nak tulis sumber produk yang sangat penting. Okay. Another product of glycolysis is ATP. ATP dalam brain mestilah ada kan? Yang ini melalui substrate level phosphorylation lah penghasilan dia. Berapa molekul dalam brain? 2. Ini 2 ATP. Okay, 2 ATP. Ini pun 2 ATP molekul. Ini melalui substrate level phosphorylation. Substrate level phosphorylation. Stage yang kedua adalah link reaction. Link reaction apa produk dia? NADH. Berapa molekul dalam brain? Obviously sama je juga, tak berubah. Kita kira ini untuk 1 molecule of glucose. Semua ni. So berapa yang terhasil? 2 juga, 2 NADH. Ini 2 NADH. Okay, the next stage adalah apa? Krebs cycle ataupun citric acid cycle. For 1 molecule of glucose kena 2 kali cycle. Okay, so maksudnya produk dia adalah berapa? 6 NADH dengan 2 FADH2 dengan 2 ATP. Okay, sama juga dengan liver. Liver 6 NADH, 2 FADH2 dengan 2 ATP. So ini adalah product of the first 3 stages of cellular respiration for brain and also liver. The next stage adalah oxidative phosphorylation. Oxidative phosphorylation hanya akan masuk NADH dengan FADH2 molekul. So kiralah campurkan. Kalau brain kosong, 2 kosong NADH, 2 masa link reaction, 6 masa Krebs cycle. So berapa? 2 campur 6 sama dengan 8 NADH campur dengan 2 FADH2. Okay. Cuba awak calculate kan. 8 kali dengan berapa? 2.5 kan? Campur dengan 2 kali dengan 1.5. Awak dapat berapa oxidative phosphorylation untuk brain? Cuba kira.
Madam.
23? 2
23.
8 NADH kali dengan 2.5 campur dengan 2 FADH 2 kali dengan 1.5 dapat berapa? 23 ke?
Ya, madam.
6, 7, 8. Sekejap. 8 kali 2 point. Berapa 3? Apa dia berkurang lah? Ada yang saya terpis out ke?
Itu
FADH. 4 FADH kan? Berapa 3? Mana lagi satu? Dah bagi kita tengok slide yang seterusnya. Ini sekejap kita hold dulu. Ni, kejap ya. Kita tengok satu-satu. Maybe saya ada tipping out tadi. Okay, NADH 2.5 betul. FADH2 1.5. So ATP yield depend on the type of shuttle used to transport electron from cytosol to mitochondria. Ini kat sini dia berbeza untuk different cell. Okay, so the electron are passed either to NAD plus or FAD. So in the liver cell, the electron receptor is NAD plus. For brain cell, the receptor is FAD. Okay, so itu akan ingat. So kat sini total net ATP yield. Okay, so kalau macam glycolysis 2 ATP tadi betul lah kan? Ini betul. Pyruvate oxidation 2 ATP. So ini kali dengan 2.5. So dapat 5. Ini masa citric acid cycle lah, betul lah. Kali dengan 2.5 dapat 5, 6. Kali dengan 2.5 dapat 15. So all akan yield 32. Ini dalam liver cell. Kalau dalam brain cell, ini awak maintain, cuma yang ini instead of NADH awak tukar dia sebagai 2, ini 2 ATP. Ini awak tukar dia daripada 2 NADH jadi 2 FADH2. Ini Krebs cycle still hasilkan 2 ATP. Ini betul. Okay. So tambah dengan 6 NADH campur dengan 2 FADH2. Dekat awak punya kertas, awak kira kan untuk brain dapat tak 30? Cuba kira 30 ATP. Ini awak kalikan dengan 1.5, awak kalikan dengan 2.5, awak kalikan dengan 1.5. Dapat tak kat sini 30? Dah kira dapat tak?
saya tak dapat itu.
Tak dapat?
Tak dapat.
Dah dapat berapa? Sepuluh. Oh, saya ni tertinggal kat sini. Kat sini ni. 2 NAD. Ini daripada link reaction. Tertinggal yang link reaction ni. Kalikan dengan 2.5. Brain dapat. So ini tambahkan lagi 5 lah. Tadi awak dapat berapa?
Tadi dapat 25.
25 campur dengan 5, so dapat 30.
30.
Tertinggal yang ni. Ini daripada link reaction. Kenapa saya kira yang ni tadi tak dapat lah? Okey, sekejap kita kira sama-sama balik. Okey. Note kat sini NADH 2.5 ATP. FADH2 1.5 ATP. Stages brain, liver. Okay, ini ATP produce. Ini ATP daripada jumlah calculation tu lah. ATP. Okay. Okay, ini stage yang pertama, glycolysis. Kalau brain, dia akan jadi 2 FADH. 2. Okay, ini awak kalikan dengan 1.5. Dia akan juga menghasilkan produk yang kedua, which is net of 2 ATP. Okay, 2 ATP untuk substrate level phosphorylation. ATP 2. Okay, ATP 2. Padamlah, cantik sikit. So ini dapat berapa ATP? 3. Ini kalau untuk glycolysis liver, dia menghasilkan berapa? 2 molekul NADH kali dengan 2.5. 2.5 dapat 5 ATP. Yang kedua, ATP yang terhasil untuk substrate level phosphorylation adalah 2. Okay. Yang stage yang kedua adalah link reaction. Link reaction akan menghasilkan berapa NADH? 2 NADH. 2 NADH kali dengan 2.5 dapat berapa? 5 kan? Link reaction untuk liver pun akan hasilkan benda yang sama. 2 NADH kali dengan 2.5. Okay. So ini akan menghasilkan 5 ATP. The next stage adalah apa? Krebs cycle kan? Krebs cycle ni kena berlaku 2 kali. Okay, ini pun berlaku 2 kali juga sebab dua pyruvate yang akan masuk. Ini 2 acetyl-CoA yang akan masuk. 2 kali cycle. So produk dia yang akan terhasil yang pertama adalah 6 molekul of NADH kali dengan 2.5. Ini pun sama. 6 molekul of NADH awak kalikan dengan 2.5. Dapat berapa? 15. Okay. 15 ATP. Ini pun sama juga. 15 ATP. Okay. Produk yang kedua untuk Krebs cycle adalah ATP melalui substrate level phosphorylation. Ini sama juga. ATP melalui substrate level phosphorylation. Berapa yang terhasil akan hasilkan? 2. Okay. So ini pun 2. Produk yang ketiga adalah untuk 2 cycle dia akan menghasilkan 2 FADH2 kali dengan 1.5. 2 kali dengan 1.5 dapat berapa? 3 lah kan? Kali dengan 1.5 ATP dapat 3. So ini je lah jumlah yang kena total up. Ada yang saya missing lagi tak? So berapa dapat dia? Sepatutnya brain 30, liver 32. 3 campur 2
30 madam.
Tadi apa yang saya salah? Saya pelik.
Tadi dekat glycolysis tu madam dekat brain tu madam tak letak tu FADH2.
Yang tadinya ni.
Yang tu.
Oh, saya tak tambah dia. Oh, okey. Ni? Tak apalah, ini ignore ya. Ini kita ignore. Yang ni je. Okey lah. So, untuk nak nampak pengiraan dia dengan lebih jelas kan, awak asingkan brain dengan liver. Lepas tu stage dia. Stage glycolysis, link reaction dengan Krebs cycle. Lepas tu setiap stage tu awak kena tahu apa yang terhasil. Glycolysis untuk brain FADH2 dengan ATP. Link reaction sama je. Link reaction dengan Krebs cycle sama je untuk brain dengan liver. Okay? So boleh nampak Nampak tak?
Nampak madam.
Okay. So itu je lah. So that's the end. Ini just nak summarize. Ini you can go through it je lah, just to summarize the 4 stages of cellular respiration lah. So glycolysis, and then kita ada link reaction that produces acetyl-CoA and then citric acid cycle. Untuk 1 glucose kena berlaku 2 kali cycle. Untuk 1 glucose juga, yang ni pun akan kalikan dengan 2 juga. Semua ni awak kena kalikan dengan 2 lah. Okay? Boleh So habislah chapter cellular respiration ni. So saya harap daripada awal chapter sampai habis chapter ni, you all faham. Ada yang orang rasa macam blur lagi tak?
So far okay madam.
Okay. So cellular respiration ni dia sebenarnya tak perlukan awak hafal banyak sangat. Just fahamkan macam mana flow cellular respiration ni berlaku. Okay? So kalau tak ada apa-apa soalan, saya nak you all sign attendance ini. And then if you don't have any question, you can leave the meeting. Thank you.